We had a few people that joined us. Anna, we're going to start with you and introduce yourself and let us know who you are, okay? Very nice, Anna. Thank you so much. Angela. We do. Yeah, we go back a couple decades together. Great to have you. Oh yeah, we go back a few years. How's that? Just a few years. That sounds great. John Altman, come on up and tell us a little bit about some of the work that you're doing there. Okay.
Well, as we started to talk today earlier about the innovation and the components around the state of Washington, I just have to say I'm a proud Cougar also. So, but a few years back in agriculture as a STEM student. Part of my piece today is around the budget and the recommendations going forward. So there's good news and bad news. We'll start with the good news of everything that happened last year, which was kind of a historic push that this alliance had made, especially around 2158 with the components around financial aid guaranteed for students in the state of Washington that are low income, and also the addition of the Washington Opportunity Scholarship, both into apprenticeships and also into other municipalities of funding sources. And that scholarship's created about 100 million on the private side and about 100 million in the public. And just to let you know, that's the only one in the nation that has done anything like that kind of scholarship. So it's kind of a gold star for everything that we do in Washington State. The other piece is career-connected learning. We heard a lot about that today, that being passed, the implementation of those grants, those regional networks, those third parties that are out there creating the opportunities for the youth. And when we look at the pieces that are there from the 16 to 29-year-olds and even others, it takes the entire ecosystem from Commerce to Washington STEM to PEI to the regional networks and everybody else that's around the tables. And we have to say thank you to two of the funders that put Career Connected Washington on the map, both the Balmer and the Gates Foundation. And that actually is being held up as kind of a national model being used across the piece. So and then the other good things that we had really looked at in 2158 was the guided pathways so that students in community colleges and bring that guided pathway down into the elementary, middle school with awareness, exposure, and then the time to degree and the right choices along the way. The computer science component, we now have a requirement that computer science must be taught in all schools across the state and also reported on. Climate science grants, the continuation of that professional development for the teachers. And within the climate area, also the clean tech and the energy components the governor is very passionate about passing last year. Continuation of FIRST robotics. I mean, you think about the FIRST robotics in the 4-H groups and the Girl Scouts and the FFA groups that Aaron has always worked hard on. There's a variety of things there. And equity supports with MESA and with other individual groups that are really looking at that. So that's kind of a flavor for all the good news. But since this is coming into a 60-day session and the budget as it was written was kind of forecasted to be about 500 million out of whack going into the supplemental. Good news is 400 million in the last revenue forecast helped that out. So David Schumacher says we're not spending a dime until I get another $100 million somewhere. Then new initiatives can come forward. But right now, I believe everybody is really focused on implementing all the good work that happened this last session. It was pretty historic. And it doesn't matter where we're at, but the technical fixes will be the first priority on the supplemental budget. And then we're not short on decision packages coming from agencies, so we're working with them. And then also the initiatives that are on the ballot may have some impact against the budget, or, well, not for the budget. Well, it could have a budget impact, and you'll see that. And then the really focus on mid-careers, because we've looked at the continuation of birth to retirement, and there's a lot of the components that were talked about workforce. Last year, CCW, or Career Connected Learning, was 16 to 29-year-olds. The governor has a very keen interest in what's going to happen to those mid-career individuals that are being potentially displaced from the workforce due to automation or other kinds of components. I think that at the packing plant today, you would see that the workforce used to be 70% manual labor. Now it's the other way around. It's more 70% technical skill and certificate and credentials. So those are some of the points that the governor is very interested in, and then also continuing his energy and climate work. And then there's a variety of pieces around keeping Washington prosperous and healthy and moving forward in the economic world. So it just, to come back to the 60-day session, we kind of predict that it'll be 60 days and done. We don't see that there would be any appetite for new revenue at this point being brought in other than what's being forecasted. And so that's more the reality of where we're at. So save your good ideas for next year. So thank you. Any questions on any of that? Okay. So I hope to make that. No, not yet. How about mid-December? Great question. Thank you.
Aaron, would you like to introduce yourself too? Good to have you here with us as well. Thank you. Jim. Okay.
Thanks, I'm back. That was a very economical presentation. You got us back on track, just about. And there was a nice teaser with that quick. So I'm Jim McPherson again, formerly director at the Tree Fruit Research Extension here. And luckily, you've gotten some prep, some background from Noel. So you've seen that WSU has presences throughout the state, every county, as well as 4 research and extension centers, one of them located right here in the Wenatchee Valley. We're fortunate here to also have co-located scientists, federal scientists from the USDA, United States Department of Agricultural Research Service. So we've got around almost 20 faculty now, right here in Wenatchee, that cover a range of program areas, broadly speaking, in agriculture, in the food and agriculture nexus that Noel alluded to. And as a research and extension center, we're embedded in our community and participatory in that community. We're also members of a larger region. And of course, our leading economic driver here is agriculture, food and agriculture. And you're going to hear now from representatives of this range of programs that do their research in various areas. Overall, scientists at the centers work in horticulture, plant breeding, plant pathology, entomology, post-harvest physiology, molecular biology, and are very important in extension. So we really are all about science-based knowledge being transmitted to and participatory with our stakeholders, not just with our tree fruit producers, but with our regional communities. And I think you'll find there are some commonalities among the researchers that are going to be presenting to you, 2 of whom are from WSU, 2 from from a USDA ARS, and then Sue Kane, our own scientist educator, is going to be presenting a synopsis at the very end. So we'll have 5 presentations, and Jean very much wants to keep us on track here. So we hopefully, the presentations will come in well under 15 minutes, and we'll try to hold Q&A until afterwards. All the scientists are going to stay through lunch. So if you do have questions, they'll be available for your interrogation. So here we go. We're going to start off with all of these folks are great researchers, early career, mid-career, end of career. They have great engagement with stakeholders. They have great teaching and mentoring capabilities, and I think their areas should be of interest to you. And again, it kind of follows on what Noel set up for us. And I think important to this group is their commitment to STEM. And as a center, we really aren't we don't have STEM programs. We have individual faculty that are very much involved and appreciate the leadership that's shown in this Apple STEM network and the MESA program. We're looking to a very exciting future and even more involvement. So I think you're going to hear some really nice stories from the individual researchers, and if nothing else, it's going to highlight their ability to communicate science in a very friendly and understandable way. I'm convinced each one of them is going to be able to do that. So we'll start off. I'm not going to do a bunch of backgrounds on the this degree, that degree, those awards. These are all distinguished researchers. We'll just come on up, tell folks about who you are, what your program is in, and then get right into your talk. Start off with Lee Kalsitz, WSU Wenatchee.
There we go. Okay. All right. So my name is Lee Kalsitz. I'm an assistant professor at Washington State University. I've been in Washington State for about 5.5 years, almost 6 years now. I am a first-generation student. I grew up in a small town of 35 people in Saskatchewan, Canada. So coming from Saskatchewan, how did I get to come to Washington State? So I grew up on outside of a small town of 35 people. My parents had a U-pick berry orchard, so that's how I got my start in horticulture. And I went on to the University of Saskatchewan. I did my bachelor's and master's in horticulture and tree physiology. And during that time, I got really interested in research and really interested in tree physiology and environmental physiology and how environment interacts with trees. And from there, I went on to the University of British Columbia in Vancouver, Canada. Dealt with traffic similar to like people from Seattle deal with. And I spent 5 years there doing my PhD and finished up in 2013. And then I started my position here at WSU in 2014. So this picture kind of showcases really what growing fruit in Washington is all about. We grow fruit in a desert. We grow fruit, and we do it for a lot of reasons. We do it because there's low disease pressure. We do it because there's low insect pressure. We do it because we have really high light intensity and cool nights that make the trees really productive, really high quality fruit, and allow us to actually have one of the biggest organic tree fruit industries in the world. In the background, you can see there's Mount Adams and Mount Rainier kind of peeking out the back. So that represents the snowpack that provides all the irrigation water that allows us to grow these trees in a desert environment that we really shouldn't be able to grow trees in. So we're reliant on that water supply from the Cascade Mountain Range to supply water to grow apples, cherries, pears, to make that the very large industry that we have right now. So when I started at Washington State, I was kind of interacting in the industry. We do a lot of that at the Tree Fruit Research and Extension Center and kind of understanding what some of their issues are. And as an environmental physiologist, these are the two things that stood out to me as some of the major issues. One being sunburn. So we grow fruit in a desert environment. It's high light, high temperature, and we're kind of, just on the edge of leading to losses. So some years you can end up with 10% of fruit loss due to sunburn issues. So the fruit gets too hot and it develops these disorders that leads to losses to the industry. And then on the right is bitter pit, and it's a nutrient-related disorder that can lead to it. Overall, across all varieties, it's about 5% of the fruit that's lost. It doesn't sound like a lot, but we have a big industry and it's a big amount. But in particular, Honeycrisp, which is the most rapidly expanding apple variety grown in Washington State, very popular with consumers, it can regularly lose 30 to 40% of its fruit due to bitter pit. So these are kind of the two issues that I've been focusing my program on. So 15% of the fruit lost, that's a $450 million economic loss to the industry. So if we can reduce that by a couple of percent, that's a large economic impact. My program is focused on developing and integrating new approaches or new technologies for solving physiology-related questions that will positively impact the fruit industry. So providing some basic science, but also being able to take that to the industry and say, these are some things you can do, or this is what's going on, and help understand their problem. So when you look at, growing fruit in a desert, we're in a high light, high temperature environment. Well, that's now. What's going to happen 20, 30, 40, 50 years from now? And when you look on a global scale, you look at changes in sunburn risk. So this is the number of days above 95 degrees Fahrenheit on a global scale. And you can see how this changes going from present day to 2080. In the Pacific Northwest, this is on a very low resolution, so you have 5 to 22 days. Other areas of the world are hit much harder. When you look at just Washington State, this is Climate Toolbox. It's a group of the University of Idaho that develops these climate models for understanding regional changes in temperature. So this is the number of days above 90 degrees Fahrenheit, which is really the temperature at which sunburn pressure starts to become an issue. It's when the fruit surface temperature is about 112 degrees Fahrenheit. So it's elevated compared to the air temperature, and then 113 to 114 is when sunburn pressure starts to become an issue. So you look at the days above 90 degrees Fahrenheit. This is a historical simulation, 1971 to 2000. There's about 20, 25 days that are above 90 degrees. You go to 2010 to 2039, so we're already into this period. And in Wenatchee, it's, I think the values are about 30 days above 90. And in 2015, we started to see some of those. 2015, 2016, we started to see some of those types of scenarios. And then you go even further into the future, so 30 to 50 years, or 20 to 50 years into the future, and you end up with more than 50 days of the year that are above 90 degrees Fahrenheit. There's a lot of sunburn pressure. So the industry is going to experience this increase in sunburn pressure more and more as we progress into the future. And it's not just sunburn or days above 90, it's also the shifting phenology or the shifting in bloom dates that can create problems. So when you look at our normal bloom date for apples in Wenatchee is end of April, early May. That's the traditional average. When you look at how this changes into the future, it's starting to shift a little bit earlier. So 2010 to 2039 is April 21st. And then we go to 2040, 2069 is April 14th in Wenatchee. Southern Washington is going to be impacted more. They're shifting almost 30 days in that period. So that shift in phenology or shift in bloom dates can create problems because earlier bloom dates mean earlier harvest. And earlier harvest, as the fruit matures and develops, it's really susceptible to sunburn as it gets ripe during that ripening period. So if it's ripening earlier in the year, it's ripening when it's hot. So it's even more susceptible to sunburn. So it's not just the temperature increases, but it's also when that fruit is maturing and when it's susceptible. So all these create some issues. And we also can look at snowpack. So we are highly dependent on snowpack for late season water distribution to irrigated orchards in Washington State. If we look at the snow water equivalent as of April 1st, this is the 2000 or 1971 to 2000 mean. On average, going through the Cascades, where most of the water comes from, is about 45 inches. And this is particularly important for the Wenatchee Valley, which grows a lot of the pears, and also for the Yakima Valley, where the water feeds out from the Cascades. The Columbia River feeds up from Canada. They're showing similar simulations, but it's a much bigger water basin and much more stable. When we look going into the future, 2010 to 2039, you see that snowpack start to drop a little bit more. Earlier melt, more rainfall in the mountains leads to lower snowpack. And then by 2040 to 2069, that overall snowpack is much, much reduced compared to what it has been historically. So dealing with those water shortages is going to be important. So my program is interested in solving these current problems of sunburn and bitter pit. We get those currently. Those are current issues that we need to solve, but also anticipating future needs and developing solutions for the industry to navigate that into the future. So I do that with a really diverse group that comes from around the world. So this is my 2017, 18, and 19 lab group. I usually have between 12 and 14 people. I have postdocs, I have graduate students, I have technicians, and I have visiting scientists and undergrad students all come in. Undergrad students come in for the summertime, visiting students come in at different points of the year, but they come from around the world. So I've had people come from all 6 continents, Antarctica excluded. I have collaborators from all over the world. Academia or research is global, and it's important to bring those global ideas. I think Dr. Schultz alluded to that earlier. So this is on a micro scale in my lab, we take that same approach. So I'm just going to kind of highlight some of the work that we've been doing to mitigate sunburn and bitter pit related issues, and then highlight some of the work that some members of the group have done. So we've been doing work with netting to reduce sunburn. This was work funded by the Washington Tree Fruit Research Commission, which is an industry-funded project, as well as the WSDA Specialty Crop Block Grant Program, which is a federal state/federal program. And during that 3-year project, I had a postdoc, Dr. Giberson Mupambi, lead that work. And he came from Zimbabwe, did his PhD in Stellenbosch in South Africa. He came and did a 1-year internship with the Washington Tree Fruit Research Commission and made a lot of contacts in the industry, and then came to my lab for 2.5 years, and he worked on this project. And during that time, we identified that netting can be an adequate replacement for evaporative cooling, which is the primary way of reducing sunburn in Washington State right now. So it's an adequate replacement. You can use netting and not have to use evaporative cooling, so you're saving water. You're saving about 30% water. And that additional netting, that reducing that light by 20%, also reduces overall water loss, water use by the trees. So it's a water conservation tool. Netting is originally designed as an anti-hail protection. We don't get a lot of hail in Washington, but it is important. It reduces sunburn by about 10%. So it's a similar effect to evaporative cooling. And since we started the project in 2015, we've gone from 1,000 acres of netting, which was basically one grower had 1,000 acres. And then now there's many growers. It's expanding. It's about 7,000 acres total of protected netting in Washington. You see that if you drive around any of the apple growing areas, you'll see a lot of netting starting to be established. And the estimated returns works out to about $3,600 in additional revenue per acre. So it's about $22 million a year impact from this type of project. So looking at understanding bitter pit and understanding what causes bitter pit in honeycrisp, this has been a kind of a fun project, understanding what is going on in honeycrisp apple that it has this problem. What's going on structurally in the fruit that causes this issue? And this has been a collaboration with Pacific Northwest National Labs. We've had time on their instrumentation there. We've also worked at the Lawrence Berkeley National Labs in Berkeley, in California, and also the Canadian Light Source Synchrotron facility in Canada. And it's all about understanding fruit structure and what fruit structure. Oh, I wonder if I can. Is there a mouse here? So what we do is we basically give medical CT scans, micro CT scans to apples. And it allows us to look at structure and vasculature. So this is a video that my visiting scientist from the Netherlands put together this last year. But it allows us to look at veining and structure of the fruit. So this is an apple fruit with bitter pit. You can see we're able to exclude the flesh. And you see this veining structure. We're interested in the density of those veins and how that contributes to bitter pit development. And we can take these types of videos and images and get quantitative data from that. And we take that two-dimensionally. We can also look at cell structure and cell density. So you look at honeycrisp on the bottom, and you look at how big those cells are. So honeycrisp has low density fruit. When you hold, especially a big honeycrisp, a large honeycrisp has really low density fruit, but you hold a Fuji, you hold a Pink Lady, you hold a WA38 or Cosmic Crisp, and it's really high density fruit. Those cells are packed really closely together, but honeycrisp is not like that. And that's what gives it its characteristic crunch. When you think of honeycrisp, it has that characteristic texture because those cells are breaking. And those cells are breaking because they're weak, because they're big. And that looks like also what's causing its susceptibility to bitter pit. So going back to the solving industry problems, well, how can we use this type of information to solve industry problems? Well, with Honeycrisp, it has a tendency to grow really big, really, really big fruit. So if we can actually decrease the size of that fruit through water management, then we can decrease bitter pit. And that's what we just finished up a 3-year project. We're just finishing up a 3-year project that was led by my master's student, Michelle Reed. She was a technician in my lab before. She started my lab as an undergraduate research intern. WSU has a research intern program where they bring undergraduate students in and allow them to gain experience. Then she came on as a tech, did her master's, and now she's back running as a lab manager in my lab. But this is a 3-year project that looked at using irrigation or reducing the amount of irrigation applied to decrease fruit size and increase the density of the fruit to reduce bitter pit. And that's been really successful. And the industry is adopting these deficit irrigation practices across the state as a kind of a common approach for managing bitter pit or decreasing the bitter pit risk in Honeycrisp. So just kind of summarize, really it's important, we're focused here in Wenatchee on solving industry problems, but it's on a global scale. We're bringing in global collaborators, we're bringing in trainees from around the world to come and work on industry problems here in Washington. And some of those examples have been protective cropping to reduce sunburn, to increase water conservation, and then also on a more basic side to understand how fruit structure leads to losses or decreases in fruit quality, and what can we do to fix that. And then challenges and opportunities going in the future that I'll highlight that WSU TFRAC here in Wenatchee is really well positioned. We're unique in that we have the ability to control. We're essentially going to be stressed no matter what. Other research areas in the United States, Michigan, New York, other apple growing regions don't have the ability to use drought as a research tool. We have that ability here, so that's a real strength. We can be a world leader in understanding how trees adapt to, and then how we can mitigate abiotic stress in the future. So with that, thank you very much for your time. And if you have any questions, feel free to find me at lunch. Thanks.
Thanks, Lee. And we'll roll right into our next presenter, Dr. Mark Missoula with the USDA Agricultural Research Service here in Wenatchee. And a sidekick.
And good morning. So, I'm Mark Missoula. I'm from ARS, as Jim said, and my sidekick is actually a graduate student in my program, Emmy Claire. She's working on her master's degree at WSU, and she's finishing up her degree in the Department of Plant Pathology, hopefully sooner rather than later. So I've been here far longer than I ever thought I would be in Wenatchee, Washington. I'm actually from Boston, Massachusetts. I did my undergraduate and my MSc at the University of Vermont. I then went to Clemson University, could not stand the South, went to WSU to finish my PhD. I then went to Kansas State University. I worked there for 3 years. Sorry, but it was the most boring place I've ever been on the planet. 14 hours to Keystone or somewhere along that lines. So ended up back in Washington State, which was actually preferable. My program has focused on the microbiome of the soil system, and overall we're trying to look at how we might take advantage of that resource to enhance plant development, plant protection with regards to eluding some of the plant pathogens that are in that system, and just improving overall plant performance. So we want to take advantage of that resource that is available to us. Unfortunately, for many, many years, it's been an underutilized resource. We know that it has value. I mean, if you think about where your antibiotics come from, most of them actually are derived from soil-dwelling microorganisms. So we know they have features, metabolic features, that we can take advantage of for human health. We're trying to take advantage of those features to enhance plant health. That microbiome is very diverse. It's composed of a lot of different entities: bacteria, archaea, fungi, oomycetes, protozoa, nematodes. And we're trying to manage that system, in a manner that improves overall plant function. Interestingly enough, some of the impacts of that microbiome on plant development may not necessarily be thought of as straightforward. I mean, who would know that the microbiome can actually influence flowering time in a plant? It influences resistance to drought stress. We know that it's very important in terms of nutrient cycling. Without the microorganisms turning that carbon over or making phosphorus available, plants would not be as productive as they are, either in native ecosystems or in agricultural ecosystems. My program has really focused on taking advantage of the microbiome in terms of managing soil-borne plant pathogens. That has really been the overarching focus of the program over these many years. So the goal is to engineer the native soil microbiome and metabolome to optimize plant health. And I think when one thinks of modifying the microbiome, oftentimes people go to utilizing a process called biocontrol, where you're introducing a non-native organism into that ecosystem and hoping that somehow or another it survives in competition with that very well-developed native microbiome to have the beneficial outcome. So if you go to a garden supply store, you can purchase various microbes off the shelf. But historically, that approach in utilizing that resource for disease management has been a failure, at least in most production systems. So we know that if we try to replant sites, so if we try to grow wheat after wheat after wheat, like on the Palouse, we tend to amplify certain pathogens in that system. We also know that in tree fruits, if we try to replant a site which has been in orchard for an extended period of time, we develop a microbiome which is actually enriched in plant pathogens. And so trying to reestablish that orchard on an old orchard site is very difficult, and typically we experience a phenomenon termed apple replant disease. Now, fortunately, we have a period of time where we can intervene during removal of that orchard and replanting that orchard. We can intervene to try to modify that microbiome. Historically, the approach has been to use a process called soil fumigation. From an environmental standpoint, that may not be so amenable, desirable at this point in time, and certainly in places like California, that's been more restrictive over the years, and Emmy's working on a project which is actually funded by the California Strawberry Commission to try to develop an alternative to the use of soil fumigation. And so what we've tried to do is intervene to modify that microbiome in a means which yields historically longer suppression of these plant pathogens than soil fumigation does. Because we know that soil fumigation, while it's very effective, its impact on plant growth is typically the first year, and those soils are reinfested by plant pathogens very quickly. So the two strategies that we've taken is to try to utilize host genetics to modify that microbiome in a specific direction. And the other is to use a soil amendment-driven approach to try to assemble a microbiome that is actually suppressive to soil-borne plant pathogens. So if we're going to engineer that microbiome, what the heck do we need to know before we actually take a strategic approach to that? Well, first of all, we need to know who is there. And while we try to find out who is there for the millennia, we have technologies today that now enable a much more comprehensive look at that microbiome. Typically, in the past, we've based our assessment of this microbiome by culture-based techniques, where we try to grow up the organisms and identify them using various visual methods. However, we know that that particular approach only accounts for probably about 2 to 3% of the organisms that reside in that system. So what we use now is a phenomenon termed high-throughput sequencing. And what this enables us to do is by targeting a specific gene which is universal in organisms, we can amplify that gene, but because the divergence in that gene is sufficient enough between organisms, we can actually identify the organism typically down to the species level. Okay, so we know who's there. What are the beneficial attributes? What kind of weapons do they possess that we might be able to take advantage of? And to explore that system, what we do is we use a shotgun metagenomic sequencing approach. And what this enables us to do is basically sequence every gene that's in that soil system. That's really powerful. It's also a real pain, because you're looking for microbial functions, metabolic functions, for instance, but you're going to pick up human DNA, insect DNA, all this stuff that you really don't want to have a handle on. And so what you do is you form a collaboration with a bioinformaticist, and you let them do it. And that's been very effective. So I have a collaborator, a bioinformaticist at the Volcani Institute in Israel. We have a cooperative BARD program, and that's been the strategy that I employed to try to get through this system. The other question that we have is, how does the altered microbiome actually regulate the plant response? Because plants have this innate ability themselves to protect themselves against many plant pathogens, right? But we know that many of those mechanisms are driven by their interaction with the microbiome. So what we do is we take an approach called comparative transcriptome analysis, where you look at the overall gene activity as it's influenced by the microbiome that's been altered by a particular strategy, whether that's the host genetics itself or through some amendment approach that we've taken. So based on all of that information, we then develop tactics that can be used to selectively amplify the desirables and limit the undesirables. So the first approach I want to talk about is the plant-driven selection approach. And what we've employed there is looking at different apple rootstock genotypes and their ability to selectively attract different types of microbial communities. And this just shows you a pictorial of the differences in the relative similarity in the microbiome from roots of different apple rootstocks. So we have G890, G41, M26, M9, and G935. These are just different genotypes of apple rootstocks. And you can see that from this figure, they're actually hosting microbiomes in their root systems that are very different from one another, although M9 and M26 in this instance are very similar. Great, they're different, big deal. What impact is this having? And so what we want now do is we look down to what's the functional differences amongst these different genotypes as far as the groups that they're supporting. And one group of great interest is the mycorrhizal fungi. Now these fungi are very important both in making phosphorus available to the plant, because phosphorus is not mobile in the soil system. One of the primary means by which plants gain their phosphorus is by making these associations with mycorrhizal fungi, which have the capacity to extract phosphorus from that system and bring it back to the plant and share it with it. The other element is that some of these mycorrhizal interactions actually limit the activity of plant pathogenic organisms. And so there's a twofold benefit if you have capacity, an elevated capacity to form the association with these mycorrhizal fungi. So in this instance, what we're seeing are two different genotypes, M26 and G890, and we're looking at the relative proportion of a group of mycorrhizal fungi that we detect in the fungal community from these plant roots. And you can see that G890, almost 25% of the what we call operational taxonomic units of that fungal community come from that particular group of fungi. In contrast to M26, only about 5% of that fungal community is represented by mycorrhizal fungi. So one would predict that G890 is going to perform much better under phosphorus-limited conditions than would M26. It also might be better protected by from some of these plant pathogenic fungi because of this elevated colonization of 890 by these mycorrhizal fungi. So that's a functional difference between these two genotypes, which we are looking to take advantage of. The other means that we're utilizing to drive the microbiome engineering is the use of an amendment-based process. In this instance, we're just utilizing a material called brassica seed meal. It's basically the byproduct of oil extraction from things like canola or mustards. And we utilize that as a pre-plant soil treatment. And what we find is that that treatment in itself does dramatically influence the composition of the microbiome. In this figure, what you see in the green dots is the relative composition of the microbiome from trees grown in the seed meal amended soils. But if you look at the control and the fumigated treatment, the black and the red dots, there's a lot of overlap there. Basically, that fumigation treatment hasn't changed the composition of that microbiome whatsoever. This is 2 years after the orchard was planted. But that seed meal amended soil has a significantly different microbiome as far as its composition relative to either the control or the fumigated soil system. Again, big deal, difference. What impact does this have? Well, that really impacted the relative colonization of tree roots by this organism called Pratolanchus penetrans. Memorize that. There is an exam after this. That's the lesion nematode, and it actually infests these root systems and causes significant plant damage. Well, this is a log scale on this particular graph, but you can see that in the seed meal amended treatment, 2 years after planting, you have basically a log order of magnitude lower root density of Pratolanchus penetrans in the seed meal treatment than either the control or the fumigated soil system. Well, what's responsible for that? And so we go back to that next-gen sequencing data and look at who's being amplified in that soil system. And what we find is that we have all of these parasites of nematodes that are being amplified in that seed meal amended system. You have things like Arthrobotrys, which is the nematode trapping fungus. It's a great thing to look at. It goes around, it creates these little lassos, catches that nematode and sucks the guts out of it. We also have Dactylella, which is a nematode egg parasite, a fungus. And we have, on the lowest level, Pasteura penetrans, which is a bacterium, which actually is a parasite of nematodes as well. So all of these organisms are amplified in our seed meal amended system, and that's providing, we believe, the long-term nematode control that we obtain in response to that particular treatment. As far as the overall benefits to the production system, in our trials, we've seen significant benefits over soil fumigation in our field trials. In this instance, we're looking at yield of Jonagold on G11. These are cumulative yields, and we see that the seed meal treatment provided a yield response that was about 25 to 35% better than the control and the fumigation. In this particular orchard, to be honest with you, the grower is a great grower, and so he really did not get a fumigation response in this instance. But it just shows you the potential, even beyond disease control, what you might be able to attain in response to this particular soil treatment. At another site, in this instance, fumigation did provide a significant yield relative to the control, but again, that seed meal amendment system provided us with about a 25% greater yield than soil fumigation by itself. So I think this demonstrates the potential for utilizing this approach to manage that microbiome, to develop a system that's more resilient to pathogen reinvasion after orchard establishment. And so now, Emmy is going to share with you, it's gone. What happened? Can you do it without your slides?
So, like he said, my name's Emmy. I'm a graduate student, so I'm going to give you a really brief insight of what my life looks like right now. So, my studies focus on dealing with developing strategies to manage soil-borne diseases in strawberry crops, and we do have strawberries in Washington, so it's applicable here. And the technique that we work on utilizes the microbial resources that are native to the system, and it's called anaerobic soil disinfestation, or ASD. And ASD is a pre-plant soil amendment, like you showed up there. It has 3 steps for application. The first step is an incorporation of a carbon source, such as like grass or molasses, and this stimulates microbial activity. The second step is to saturate the soil with water to fill pore spaces, and then the third step is to tarp that treated area to limit gas exchange. So ASD has provided control of soil-borne diseases across a variety of cropping systems, and it doesn't face the same restrictions or health concerns as chemical fumigants, so it's a really promising technique. It also has the potential to control even more diseases with some optimization. So we know that the research says that the efficacy of ASD is highly dependent on the carbon source used as well as the target pathogen. So to better understand that relationship, we designed an experiment to evaluate the efficacy of different carbon sources in conjunction with ASD to control fusarium wilt in strawberry. There we go. That's kind of what ASD looks like in a very cartoon manner. So yeah, we have this experiment. We're testing different carbon amendments. And our ultimate goal is to link the changes in the soil microbiome and metabolome to resulting levels of disease control. So what we did is we chose fusarium wilt. It's a notoriously difficult disease to manage. We like a challenge. And then we tested 3 different amendments. We tested rice bran, orchard grass, and wheat grass in conjunction with ASD. And we found that none of these amendments resulted in a completely suppressive system, but we did notice that disease symptoms were delayed when compared to the no amendment control. So to investigate why one amendment works better than another, we designed an experiment to characterize both the chemical and biological components of the soil over the course of ASD treatment. And we did this utilizing high throughput DNA sequencing to characterize the soil microbiome, and then various analytical techniques, including GCMS and LCMS, to characterize the soil metabolome. We use this approach because disease suppression achieved through ASD has been attributed to the production of metabolites that are generated by soil microbes during the ASD process. So our ultimate goal for this, because the soil metabolome hasn't been fully characterized yet, was to document the changes in the soil fungal and bacterial communities and correlate those changes with the production and consumption of metabolites over time, with the idea that hopefully we can pick out key organisms and compounds that might be involved in disease suppression. And what we found was that our target pathogen, while it wasn't suppressed in the initial stages of ASD treatment, it did show a general decline in its population over the course of the entire treatment. And that corresponded with increasing populations of certain bacteria, particular anaerobic firmicutes, and also increased production of metabolites with antifungal properties. So we needed to do some more work with this. It's not completely finished. We want to evaluate the functional role of specific organisms and compounds in regards to long-term disease suppression. But doing that will allow us to make better decisions when we're optimizing treatments like this, not just for fusarium and wilt, but for all soil-borne pathogens. Thank you.
Great professional training for things mess up on you, Emmy. That was really nicely done. And Mark, glad you moved from Kansas and stayed here. Up next, another USDA agricultural research scientist, Lauren Hannes. Lauren will describe where he comes from and what he does.
We're going to back up here a little bit, I think, so you can see everything several times. Just close your eyes. Okay. Right, so my name is Lauren Hannes. I'm a molecular biologist with USDA ARS. I came to tree fruit from parasitic plants. So I got my doctorate at Penn State, where I kind of bridged the gap between molecular biology and bioinformatics. I'm trying to study and understand parasitic plants, how they become parasites, and how to control them. The worst weeds in the world are parasites, and so we had kind of a big goal for us. But then I realized that there was sort of limited capacity to transfer what we were doing with parasitic plants to the people that actually needed it. I wanted to get a little bit closer to applications. So that's kind of what led me to looking in tree fruit research, is that we have a much closer relationship with industry partners. So what I'm going to talk to you about today is my work and sort of my vision for how we can ensure or enhance fruit quality using biomarkers. And a biomarker is kind of like a lipid panel when you go to the doctor, combined with your family genetic history to predict your risk for heart disease, right? They're looking at biomarkers to figure out where you're going to be in the future, okay? You want to do the same thing for fruit. So here's a lab where we work, Snowy Mountains in the background. Where there are we're 5, soon to be 6 scientists at the ARS Tree Fruit Lab. We're kind of split into 2 groups. Mark's group and other scientists in that group focus on pathology. The other half of us, Jim Mathias, Dave Riddell, and I focused on post-harvest fruit quality. What do I mean by post-harvest? Well, I work primarily with apples. I have a few pear projects, but really post-harvest is about maintaining quality during the post-harvest period, right? So whenever you get a piece of fruit in the store, we want it to look nice and we want it to taste as nice as it looks, okay? There's a lot of technology that goes into producing fruit that looks like this, right? You don't really get to see much of the bad stuff. Right? They take that out of the supply chain before it gets to the store. So this is a snapshot. It was in an NPR article, and this is just showing you the inside of a typical storage shed where you can store about 10,000 pickup truckloads of apples or pears up to about a year. And there's a tremendous amount of technology at play to get to that product, right? To get to a nice eating piece of fruit after a year. Our storage chambers are a little bit smaller. We have about 150 of these micro chambers at our research unit. But these things can do all the same things that the big ones can. This includes controlling temperature. It's pretty typical to store apples just above freezing. We can control the atmosphere or the air inside these chambers to limit or manipulate oxygen and CO2 in order to prolong the shelf life of apples. We can add hormone blockers that basically help the fruit hibernate for long periods of time, that slow down their ripening so they remain a nice eating piece of fruit for a long period of time. We can add antioxidants to reduce oxidative stress. There's even real-time monitoring of fruit stress that, for instance, and I'm not going to go into the details, relies on chlorophyll fluorescence, right? Apple peel does have chlorophyll in it. We can also look at fruit respiration. So just like you and I are breathing in oxygen and breathing out CO2, fruit are doing the same thing. So all those beautiful pieces of fruit that are there on your desk, when you take a bite out of that, I want you to remember that thing is alive and happy, and that's contributing to your good eating experience. So I'm really fortunate because I'm coming to post-harvest with a wealth of information that's been produced over decades of work, and a lot of it's come from the ARS and WSU right here in our valley. We know a lot about post-harvest physiological disorders, and I want to stress here that this is not the result of pathogens. These are problems with the fruit themselves. We know a lot about how to provoke these disorders, a lot about how to prevent them. We know which cultivars get them, which ones don't. What we don't know is why, right? So why, so why does this part of the peel turn brown and this part does not, right? We don't really know much about the mechanism, and that's what I'm after. I want to understand the molecular mechanisms of post-harvest disorders, because if we know more about the why, we can do a better job of controlling these costly disorders. Like Lee said, it's to the tune of millions of dollars a year. So to do this, we turn to plant genomes. So what is a genome? A genome is a collection of all of the genetic material in an apple. That includes genes. Genes are instructions for proteins, and proteins do basically everything for us. So they're the enzymes that are digesting our food. They are the crystals that make up the lenses of our eyes. Here's a fun fact for today. So plants also have these eye crystal proteins, but they use them for a different thing. This happens to be a really ancient protein family. It's shared between animals and plants and fungi. There's your fun fact today. Okay, so plant genomes are really complicated places. And this illustration is from a paper about 1.5 years ago for the Golden Delicious genome. And what it shows you is each one of these segments, as we go around clockwise, each one of these is a chromosome. And across the genome, you can see that each chromosome has a genetic twin in another spot. Okay? We take this as evidence that at some point in the history of apples, there was a whole genome duplication. So some ancestor duplicated all of its genes and then carried on as a new thing. And the evidence here suggests that this happened about 50 million years ago. But what this does is it makes the puzzle really, really complicated, because there's a lot of spots in the genome that are similar to a lot of other spots in the genome. And when we consider that the apple genome has about 600 million letters of code, several tens of thousands of genes, it's a really, really complicated place to wade around in. But we're going to try and use it in order to develop strategies to produce consistently good fruit that has a nice long storage life, and try and come up with ways to limit these issues during the post-harvest period. So when I was a graduate student, I got to graduate school in about 2005, and after I finished up rotations and coursework, the second generation sequencing revolution hit. And it went from costing $10 million to sequence a genome to just a few $1,000s. And so around this time, I figured that I'd better learn some bioinformatics because the wave was coming, and it's a tremendous amount of information. In public repositories, we literally have tens of trillions of these little pieces of DNA letters of code, and there's more than this coming all the time, and this is doubling faster and faster. So it's literally a flood of really, really big data. This was afforded by a machine created, or by a technology that Illumina bought early on from a company called Solexa, which is not really important. But the ancestor of this machine is really what drove this revolution. The latest iteration of this machine can sequence 3,000 apple genomes in about 72 hours. There's a catch, though. Everybody's like, wow, great, there's a catch. The catch is that this machine, even though it produces 3,000 genomes, it's in a lot of really, really tiny pieces, like hundreds of millions of tiny pieces, okay? And so the puzzle is still really hard to put together. And part of the reason it's hard to put together is because the genome is a really complicated place. Remember, there's duplications, sites that are really similar to each other. So one of the ways that we can make the puzzle easier to put together, like this one that's a few thousand pieces, is make the pieces big. Big pieces are still really super expensive, so we can't do that. The other way that we can do this is we can create a picture for each puzzle, right? So if you ever tried to put together a really complicated puzzle, how many times have you looked at the picture to try and figure out where the piece with the leaf goes, right? It's a really important strategy. So what my lab is aiming to do is to generate reference pictures for each of the genomes, right? We need a picture for each puzzle. We need a genome for each cultivar. And that's because, and this is an experiment that we did in my lab with a collaborator from Cal State, where we scanned the Granny Smith genome for differences. There are 5 million differences across the genome, which makes Granny Smith more different than Golden Delicious than human is to chimpanzee. So imagine if all of our human genomics and biomedical work was done leveraging the chimpanzee genome, right? There would be some problem areas, right, where chimps and humans are really different, right? Mostly the same, but the crucial bits would be different. And that's what my lab is discovering, is that the things that make a Granny Smith unique are the exact blind spot that we have for these large-scale technologies. So here's a quick snapshot of my team. So there's folks here at the USDA who are working here in my lab and in my research group. I have a number of collaborators at Washington State University, both here and in Pullman. I've got collaborators at Penn State, at Cal State Fullerton, and a lot of the work in my lab is supported by the Washington Tree Fruit Research Commission and then base funding from the agency. We're a multidisciplinary team. We use bioinformatics, horticulture, physiology, and molecular biology to build genomes and then use those genomes to interpret really large-scale data to try and tease out what genes are important and understand about physiological disorders of fruit. So I'm going to talk to you a little bit about an experiment that we've done recently, and hopefully you'll see after this kind of how we're aiming to get biomarkers out of this information. So this is the green apple, the one, right, Granny Smith. We picked these fruit and treated them early in the storage period with a couple of different things to prevent a disorder. So the control fruit received no treatment, and then there's a couple of different treatments here. One's organic compliant, one's conventional compliant. But even after 4 months of storage, all the fruit looked the same. Okay, but in these two, we treated them to prevent an eventual disorder that you'll see here in a minute. So 4 months go by, 5 months go by, 6 months go by, everything great. But when you pull the fruit out of storage and simulate a supply chain, when you wake these fruit back up again from being at just above freezing, this happens. And it's kind of hard to sell those fruit, right? So we'd really like to know, okay, when is this going to happen and how can we prevent it? And like I said, we know about how to prevent some of these disorders, right? We're able to do it, but we don't really know anything about the mechanism, okay? In order to push the envelope a bit further and do a better job of controlling it, we need to understand the why. So it's going to get a little messy here. Okay. In the trade, we call this a radiculogram because it's kind of meaningless to look at. But bear with me. I'm going to walk you through it, and I'll tell you exactly what it's showing us. So in our experiment, in the first week of storage of these fruit, we gave the fruit a couple of different treatments and the control. 2 of them in the first week that received this treatment didn't get sick after 6 months of storage. So something really important happened in that first week or 2 when the fruit were receiving that treatment. We generated samples where we analyzed the activity of all 45,000 apple genes. We took a little over 500 million measurements, which we need computers and coders to analyze, right? And so we distilled it all down to about 1,000 genes that we think were really important. And each one of those 1,000 genes is a dot on this slide. Okay, so each gene is a dot. When the fruit were getting a treatment that prevented them from getting sick, the blue genes were being used less, and the red genes were being used more, okay? So for instance, this collection of red genes right here were being used a lot by fruit that were getting a treatment that weren't getting sick. And so when we try to understand what prevents this, right, we can look at the functions of these genes and try and figure out what was happening in that piece of fruit that led that treatment to be successful, that stopped this peel necrosis. Okay? Now remember, I was talking about genomes and the puzzle, so we're going to come back to that. the critical thing to remember here is that this puzzle is probably incomplete. And so the very things that make Granny Smith special, maybe the genes that make Granny Smith a cultivar that is uniquely susceptible to this particular disorder, may be in the blind spot of our analysis. So to address this, my lab has sequenced the Granny Smith genome, and we're now in the process of annotating it, and we're going to go back and reanalyze these data and discover which pieces of the puzzle are missing. Because what my lab has done so far is figured out that these might be the most important genes in this process. So now I'm going to switch gears a little bit, no more ridiculograms or pictures of genome plots. And I'm going to talk a little bit about the people that are in the lab. So this is a snapshot of the junior scientists that have come through my lab in the last 3 years. And some of them are here from Wenatchee, some are in remote places, some of them are from collaborations with Lee Kausitz and Mark Missoula, who you've already heard from today. So Sumaya was a postdoc in Lee's lab, and she came and worked in my lab. We trained her and worked together on a project where she learned a little bit about bioinformatics and molecular biology. We did help do some data analysis with Shashika. Sophia is in between high school and college, and she worked with a prof at the WSU TFREC, a new person, Carolina Torres. Elena was the first undergrad that came through my lab, and now she's a college graduate, and she's a technician at the Fred Hutch Cancer Institute, and she's looking at going to grad school. Becca, who came by way of recommendation from Sue Kane, worked in my lab and did some molecular biology, some bioinformatics. Bruno did some molecular biology this summer. Lindsay Hildorfer is actually a local teacher, another recommendation from Sue Kane, who worked in my lab this summer. There are a couple of students here who are supported by a project in my lab, but through a collaborator at Cal State. They are leaning more towards the bioinformatics and computer science end of things. At Penn State, Eric Wafulo does a lot of the heavy lifting in terms of genome assembly and genome annotation. John Haydes is a graduate student that I advise. His major professor is in Pullman and also in bioinformatics. And so I've been really fortunate to have a great selection of students and junior scientists come through my lab, but where they come from is a lot of times a pool of people here in the Valley. Like I said, about half of these folks came from Wenatchee. And through collaborations or partnerships with WVC, the MESA program at WVC, through the Apple STEM network. And so we work really hard to build these partnerships in order to keep the stream of really talented people coming because, man, we need them. Part of that is through outreach. Typically, the mission of outreach in ARS has been focused on stakeholders and industry folks, and that's still the core of our outreach mission. But recently, that mission has expanded a little bit to come sort of more in line with my ambitions for doing outreach, which is good because now my activities also line up a little bit better with my duties. And in doing that, we've interacted with a lot of students. And what this has basically grown into is a biannual event where we bring students to the lab from Orchard Middle School with partner teacher Beth Hammerberg, and we talk to them about entomology, physics, disorders, robotics, data analysis, microbiology, molecular biology. They get exposed to a lot of stuff. We've interacted with about 500 students in the 3.5 years that I've been here at ARS. And a crucial component of that outreach activity is leveraging volunteers that come from local STEM organizations. And so we've got graduate students, people from WSU, people from MESA, students, parent volunteers, people that are really getting involved and engaging these students in STEM. And so hopefully, and even a little bit selfishly, we want to keep the stream of STEM interested people coming through the Valley and getting involved in research because it's good for the health of research here, which means it's good for the health of the industry. And with that, I will end, and if there's time, I'll take a question, but if not. No questions allowed? Okay, we'll wait to the end.
Next presenter, Dr. Kate Evans, is going to regale us with the story of apple breeding in Washington State. Kate's now the interim director at the Research Center and Extension Center here in Wenatchee, as well as professor in the Department of Horticulture.
Are they at the beginning of mine? Oh, okay. No, that's okay. Okay, great. Who am I and how did I get here? Good question. So I'm English. Okay, that's the accent. It's not Australian. Just setting you straight there. Common misconception out here in Washington. Okay, I did everything in the UK kind of before I got here, but I've been here about 11 years. I did all my education in the UK. I had 4 children in the UK. I'm not sure I would have had 4 children regardless of the issues of the joy of raising children and having a STEM career. I think there are certainly still challenges there, but having my 4 children in a country where I got 6 months maternity leave, whether that meant that I brought my children into the field while I was selecting from my seedlings, that's different. But anyway, so yeah, I came here 11 years ago, based in Wenatchee, the home of new apples for Washington State. So what I do is really run the breeding program. It's a team effort. So I've listed current team up here, jointly funded really through WSU, but also for many years from funding from the Washington Tree Fruit Research Commission, so grower levy money. So what is the aim of the program? The aim of the program is to get a portfolio of new improved varieties for our state growing conditions, really important, and that those varieties are actually available for our growers. So you set the scene for the breeding program. The breeding program didn't start until 1994. Seems like a long time ago now, but in apple breeding terms, that's relatively new. And really what we've been, it was based on the fact that varieties that are grown here are not selected for our growing conditions. I mean, we're doing a fabulous job growing those varieties. I mean, you've got Honeycrisp sitting on your table. It was selected in Minnesota. You don't get very much different really from growing conditions between Minnesota and Washington. So if we're doing that greater job with varieties that were selected elsewhere, what could we do with varieties that were especially selected for our growing conditions? And then when you go back to the 90s, many of the varieties that were actually released under an exclusive license, which meant that many of our growers just didn't have access to those varieties. So there are a lot of new varieties that came in where only some of the bigger players in the industry could actually access them. Okay, so apple breeding program aim. What I'm going to do really is tell you the Cosmic Crisp story, because I've found over the last couple of years that whenever I come and give any kind of talk, all the questions are about Cosmic Crisp. So I'm just going to tell you the Cosmic Crisp story and try and put a 20-year story into 5-10 minutes, maybe. So you've heard a little bit about Cosmic Crisp already, or WA38. Various people have alluded to it. Dr. Schultz showed the mother tree. We were out there paying homage to the mother tree on Monday, which was a kind of very cold but fun event. The Cosmic Crisp is a result of a traditional cross between Enterprise and Honeycrisp that was made in 1997. It is a fabulous apple. If anybody's tried it yet, hopefully everybody's on the same has the same opinion. Okay, there will be people that don't like it, because there are people out there that, no, I can't start to talk about negatively about other apple varieties, but you know, everybody has different tastes. Okay, if you like Granny Smith, that's okay. But importantly for our industry, it is a variety that has got exceptional storage potential. So you know, we're an industry that is based on producing this massive volume of fruit. We want our fruit to be able to store for a long time so that we can market fruit for 12 months. Okay, so going back to a little bit about the Cosmic story itself. So it's from conventional breeding. Most people, it's a common question. Oh, it's a new apple, it must be GM. Okay, it's basic line of questioning for me. No, I mean, what we've done in creating this apple is technology that's been around for about, in this form, for about 300 years. So it's really not modern. Kind of pains me to say that, but it's really not modern. It's all about, fundamentally, it's all about sex. Okay, so that's a good hook to STEM education for certain age groups. Puts others off, but you know. So what we're talking about is taking bloom, taking pollen from bloom. In the case of Cosmic Crisp, it was Honeycrisp pollen, and then putting that pollen onto stigmas, the female part of the flower of Enterprise. And here's our little vial of pollen. We can use a paintbrush or a pencil eraser or whatever. It's really not high tech. But what we want is we're making that cross, we're making that cross to produce seeds. And the seeds are the things that really we're interested in. These seeds are, for this particular example, for Cosmic Crisp, these seeds are a combination of their maternal parent, which was Enterprise, and their paternal parent, which was Honeycrisp. And now they might all look the same, but every single one of those seeds is different. Okay, and so the best way to think about that is if you look at your family, you have your mother and your father, and you've got your siblings. Okay, well, your siblings are all related in the same way to your mother and father as you are, but they're all different. Okay, and the same kind of things that segregate in your family kind of segregate in our apple populations. So there are a lot of things that are easy to see that are segregating in there. And when we think about apple, it might be skin color or vigor, size of the tree. You know, when you think about humans, we think about hair color, eye color, nose shape, your ears stick out, that kind of thing, all those nice family traits. So these seeds might all look the same, but in fact, every single one of them is different. And the breeder's job is to select the best individuals from our progeny of seeds. So with the Cosmic Crisp, here's a sort of continuation of our story. The seeds from that cross were germinated, and this is just a shot of those seedlings in the greenhouse. Of course, they all look the same as well. We're still not starting to see differences. And we don't really see differences until we plant those trees out in the ground. And for Cosmic Crisp, it was planted out in the orchard in about year 2000, very closely planted along with its siblings. So all of those have got the same mother and father, but they are different. And really, we start to see those differences when we harvest the fruit from those individuals. And we do a lot of testing in terms of instrumental testing, just kind of pressure testing, juicing, looking at sugar contents and things. But fundamentally, a lot of the testing that we do is quite, I'm not going to do this, but it's quite simply just kind of taking a bite out of your apple. And, you know, there's nothing yet that is as good at evaluating an apple as we humans. So in terms of the Cosmic Crisp story, we evaluated it at this phase where there was just a single tree, and that was the tree, in fact, that we all went and paid homage to on Monday. We then decided that it was a good enough tree to multiply and to put into a few different sites. We used clonal propagation, that is what the industry at large uses to make more things that are the same, taking a bud and putting it onto a rootstock and making a new tree. We put it into 3 different sites. In Washington, here's Wenatchee. We've got a site up on the banks of Lake Chelan. It's a cold, late harvest site. And we have a site down near Prosser, which is a warmer, earlier site. And we put 5 trees in each of those sites, really to start collecting some decent data, because we always want more fruit. We always want more data to determine whether or not a variety is worth moving on with. Well, obviously, from where we are now, it was worth moving on. So the decision was made to propagate it into our 3rd phase of selection. Here we are really looking at trying to get large volumes of fruit. And when I say large volumes of fruit, it's really so that we can do these kind of tests. We can run that fruit, those selections down a packing line. We can see whether it will handle a packing line. Will it work in a commercial type situation or not? Here you can see this is our phase 3 orchard. We're getting a lot of fruit. We can see whether a piece of fruit will wax or not. Importantly for this industry, fruit is waxed. Some selections don't hold wax very well. It's kind of just a function of the skin. And we can do some getting that fruit out to consumers. Really important. How does it taste? What do people think? We first did a consumer test of the WA38 or Cosmic Crisp apple versus Gala back in 2012. And regardless of what people say, we put out fabulous quality Gala, and it wasn't rigged in any way. And here's our consumer preference. The blue bars are all the WA38, and the red is the Gala. We got great results. Obviously, it's looking really nice. Well, I showed it to the industry, and their comment was, yeah, well, that was Gala. What about Honeycrisp? So we did it against Honeycrisp, and it was really good. So that was kind of a deciding factor of the whole process. Okay, so then, having got all of this data, there was a decision in 2013 to release the Cosmic Crisp apple, or WA38 as it was at that point in time. Then it got difficult. How do you release an apple? It's not easy. A lot of discussion with the industry, a lot of discussion within WSU. Where we are now is that apple has been released as a Washington-only apple in the US for 10 years. Only really fair because Washington growers put in funding to fund the breeding program for many years. So, hey, they get a head start. That's how it should be. We had about 600,000 trees planted in 2017, another 6 million trees planted in last year, and about 5 million have gone in the ground this year. And the first fruits should be available for sale this year, December the 1st. There is a lot of hype about it. I'm kind of hoping that some people have heard of it by now, particularly in Washington. If not, who are you connected to? I mean, it's out there on every bit of social media that you can find. And really that's because WSU partnered with a company called PVM, or Proprietary Variety Management. That's a really hard one to say, but PVM. To build the brand, because you can't release an apple these days without having a brand development. People buy apples by name. They get to know a name of an apple. And how are we going to entice people to buy one of these many apples that we're going to be producing over the next few years in Washington? So there is this big campaign out there at the moment. If you haven't checked it out, just search Cosmic Crisp and you'll find all sorts out there. We even have an astronaut who visited the state this week to do a bit of the influencing. I'm learning the kind of terms, right? Anyway, what's next? So people ask me, okay, well, hey, you're done now, right? Not really. The breeding program keeps moving. I was actually quoted to say that we have a conveyor belt of stuff moving forward, which was not very technical, but we have a conveyor belt of new varieties moving forward. So there is a lot more. If you remember my initial aim, it was to produce a portfolio of new varieties, and there is certainly a lot more material in the breeding program yet to come out. So watch this space. More science, more development of breeding science. Although I said the technology that we're using currently and did use for Cosmic Crisp is about 300 years old, a lot has moved forward in terms of our breeding science, our molecular science, the use of DNA markers, etc., in our selection. Okay, we're here in STEM. What's next for me also is more new scientists and plant breeders into the workforce. I'm a public plant breeder. It's kind of a passion, really, is to educate more plant breeders into our workforce. Plant breeding is the thing that has produced most of the food that you're eating. Most people are not aware of plant breeding. I mean, it's, anyway. So this is my current and previous student team based, just to kind of show you a bit of diversity from my current postdoc, current grad students, previous grad students, research experience undergraduates, current intern, a sort of wide mix. But I think also what I wanted to say is that I'm also trying to pay it forward. So what I want my students to do, my students get very involved in this, is doing more STEM out into the community. So we've hosted multiple visits. We've had Evergreen College, we have Wenatchee Valley College Post-Harvest Group on an annual basis. We've a few years back now, UW got in touch with us and said, hey, we've got this great STEM grant, Gear Up. It's all about focusing on Yakima Valley, and we want to take out some science and STEM stuff out into the Yakima Valley, but we suddenly realized we don't have any agriculture to talk about. So can you come and do that? So we did their Gear Up, these two, which was the whole, I mean, it was a massive load of middle school, predominantly kids came through that one. Spent the day standing next to a giant inflatable colon, which is the first time I've ever done that. Hopefully the last. And then also this picture at the bottom is just to remind me that one of my students last year went and taught a genetics class up in Leavenworth, 8th grade genetics class. Fit their syllabus perfectly. They were doing genetics. We thought, okay, let's talk about apple. Sue linked us in, and so. What better thing to do to talk about apple with genetics in the Wenatchee Valley? And in fact, that same student came back from Minnesota, where she's from, last week and said, hey, guess what? I had to go and teach an 8th grade class in Minnesota on apple genetics. So that was kind of nice. So yeah, more STEM. For me here in Wenatchee, I have 4 kids. I reached out to Sue earlier in the year because it was a big issue for me. My kids go through the Wenatchee School District. No AP Bio this year, okay? No AP Bio. So yeah, we've got computing. We've made some moves forward, but no biology? Really? So we're doing it online, okay? My kid is fortunate, perhaps, in the fact that he's got 2 parents who are biologists, so we can kind of push him in that direction, but yeah, we've got some way to go, I think. Thanks.
I'd like to thank all 4 of our presenters, great researchers, great mentors, with a real commitment to STEM. And now, to wrap it up and put it all into one synopsis, along with their backup group, the Galactic Farmers, here's Sue Kane.
Hi. Yeah, so I had the task at first of maybe talking to you a little bit about how you can now imagine that apple sitting in front of you as a newest symbol of the tech industry in Central Washington, because you now have some great understanding of the apple as a product, and from conception and the breeding and the genetics programs and all of the work that's going on there. If you think about the 20 years of work to get that from from a seed to the product line, if you understood how about some of the work in the soil amendments and being able to create an environment that produces more fruit, that helps that fruit be resilient to infectious disease, and increases the productivity of the industry, to the post-harvest disorders and some of the work that's being done to ensure that it can not only sustain storage, but also the distribution. And really, the first couple of times that I started to think all the way through what it takes to get something from the field to the grocery stores, it really sunk in that I had no idea about all of the research and the tech. And I'm a scientist myself. And when I first moved to Wenatchee, Washington 8 years ago, I had a PhD. And I moved here with my spouse. He's from East Wenatchee. And actually, I put this up, this slide, because one of the first things that he ever said to me was, I'm from Wenatchee, Washington. It's the apple capital. You may have passed this sign on your way in. And I kind of smile at this now because even knowing that this was the apple capital, I didn't identify it readily as a science community. As somebody coming into the area, I didn't know about the technology and the research. I was a scientist myself and in the habit of seeking out that kind of camaraderie and intellectual opportunities to kind of dive into data and into theories with other scientists. And I really leaned in towards the community college, which is where I started working when I first got here, and found some really great colleagues that fed that. But right down the road, there was this WSU research center, and there was a USDA lab, and there were scientists engaged in research that I found fascinating. And maybe it's a better thing that I've said this before. Had I known about Mark Missoula's research in soil amendments when I first got to Wenatchee, my trajectory might be entirely different. But I think now that I know about all of that, it became one of these really kind of personal missions. As I saw some of the students come through my classroom at Wenatchee Valley College, and they also don't necessarily realize that there are these opportunities for them in STEM that make use of their identity, their background. And so I have an opportunity here, and I'll introduce them in just a second, to highlight some of those students in our region that we really want to see students be able to take the opportunity of who you are, where you're from, that agricultural background, and some of the ambitions that Noel was talking about this morning. They are humanitarian. They want to change the world. They want to be able to innovate and be creative, and that is why we are all in this room. And they can really do that if you think about the opportunities through food production and the opportunities to really kind of look and innovate in the face of climate science and climate change. They can be empowered through agriculture to be the scientists and the technical data scientists, the computer statisticians. We'll learn a little bit more about some of the Wenatchee STEM industries, or the North Central Washington STEM industries, rather, this afternoon. But I want to set this up just a little bit before I bring up the galactic farmers for you to meet. And I want to tell you a little bit about one of the ways that we've been working as a system together to tell our students in our community about these future careers. Today, one of the options, you have highlight tours that you can take at Stemilts or at Rocky Reach Dam. But the third option that you have is to stay right here in this room and to take part in one of the community features that's been, for the last 2 years, really instrumental at catalyzing some of our regional collaboration. We are in North Central Washington. We have 2 of the community colleges that you heard from this morning. We don't notably have a 4-year in North Central, although Central Washington University has been a fantastic partner and has come over for a regional collaboration. And we've been doing some work to build some pathways, and we'll talk a little bit about that this afternoon. But with the absence of that really strong 4-year presence locally in the region, what we miss is some of the benefits that that 4-year ecosystem brings. The networking and communication and visibility of those developmental pathways, the college-going culture and mentality of some of the generational, even the cadence. I went to graduate school in a small town where school being in and school being out kind of defined how that community was operating. Those kinds of things are missing largely from some of our rural communities here. And so it was over some conversations with Jim McPherson and Lauren Hanas and Jim Mathias at the USDA and Kate, we kind of started to talk about how do we create a better visibility about the types of research that are happening in the region. How do we start to cross-pollinize the student populations, whether they are K-12 or undergraduates at Wenatchee Valley College, or they're the graduate students. It is an important part of a graduate student's development to have regular opportunities to communicate and mentor. And the students that are here at WSU, they miss that often. They need that opportunity to go visit 8th grade students and talk about apple genetics, right? So we had an idea that is not a new idea, but it is new outside of a 4-year institution footprint. We started a seminar series, and it is tailored to us. It runs in the fall for about 8 weeks. It runs in the spring for about 8 weeks, and that makes it possible for our scientists that are out in the field over the late spring to early fall timeframe to highlight their work. And we work with the local media and the STEM network and the Technology Alliance, the Our Valley, Our Future, which is a local public-private grassroots organization. They've helped us to really get the message out to the community about the vibrant research community that's here doing that work. And so when we put these together, these are science seminars. They are our local scientists taking time to do that outreach, to present to a very mixed population. You may have a geologist sitting next to a K-12 teacher, sitting next to an undergrad who's getting extra credit for being there, sitting next to someone who's a postdoc and has a breadth of experience in tree fruit research. And the collaborations and the synergy that have come out of those networking opportunities, I know I don't have to tell many of you the value of those, but I think monetarily we will actually start to see new dollars for research collaborations coming in. We're certainly seeing the exchange of the workforce development pipeline. I've seen several students advance from WVC into some of those labs and seen even some of our teachers taking opportunities to do some research in those labs. So today, we actually get to highlight the research of another one, if you haven't gotten your dose of science yet. This one is Dr. Vincent Jones, and his work is really interesting because it will use, he uses the data analytics and the computer computational power of the computer to really help growers understand when a integrated pest management strategy might be most beneficial to their crops. And so it again kind of infuses some of that data science work with agricultural practices. And this is where that industry is heading. This will have global significance, and it's happening right here in the region. These seminars are open to our entire community to take part in. And so it has been going out in the newspaper, and it was on the radio this morning as I was coming in. And I love that. And so if you are interested in participating in that today, it will be in this room, actually. So you can stick around here for that. So I'm going to segue here, and I'm going to tell you one last thing about another one of our local keystones in our STEM ecosystem, the Greater Wenatchee Area Technology Alliance, GWATA. I actually co-direct the STEM network alongside Jenny Rojanosatian, who's in the back of the room here. And Jenny has, the STEM networks all across the state of Washington have a business component and an education component, and they are arranged uniquely in those 10 regions. No two are alike, which is great. Here, the education, I am at the North Central Education Service District and have the ability to bring some of the school districts and higher ed educators to the table. And Jenny, through her work with the Technology Alliance, is able to bring opportunities for business to engage in STEM education. So together we are able to put together a huge portfolio of opportunities and synergistic initiatives, we'll say. One of those initiatives was just last week, and the GWATA is the Greater Wenatchee Area Technology Alliance. The GWATA Innovator Awards last Wednesday is an event where about 360 of our community stakeholders come out for lunch and recognize 5 awards. And those awards go to a entrepreneurial business of the year. They go to a future technology leader, which is a student award, the post-secondary student award, post-secondary innovator of the year, which I think President Richardson was talking about. Wenatchee Valley College took home again this year. And I'm missing the others, but the award ceremony, these are the winners of the award here recognized on the slide. But the K-12 Student of the Year award last week really embodied the spirit of what I was going to try to talk to you about. So I thought rather than standing up here and telling you about it, I would introduce you to some of the students that we're hoping to be able to build these pathways for and hoping to be able to continue to develop their love of agriculture and their identity around it, and you'll see that here in just a second, and technology and where they're able to incorporate that learning. Their teacher and their superintendent are here as well today. And as I ask them to come and join me up here and I'll introduce them, I want to play you the video that highlights their award. Let's see if I can get this to work.
The Galactic Farmers is a STEM team that was put together by a bunch of friends to participate in robotic competitions.
Our project was the Apollo 50 Angles Challenge. We had to recreate what Apollo 50 mission would be like on the moon.
You fly a drone and land it on a target with your payload, and there's a EV3 system, and you're supposed to come put the payload down, and then the EV3 pushes it into another target, and then you get a whole bunch of rock samples and then bring the rock samples back.
In the rules somewhere it said represent your community, and we took that to a whole new level. We are very proud to be farmers here.
In a small community, it feels like you're kind of boxed in, but if you expand, like think outside the box, you can do a lot more things, which the normal world usually does. So it's cool that we have all these opportunities presented to us that we can try out.
This project seemed really special to us because we had an opportunity to show off our community, to show that even our community being a farmer town, being small, we could still go out and do huge things.
I didn't know we would get as far as we did. I feel like the first week we were here, no one knew what they were doing. A couple weeks later, we all just clicked into position and we knew what we were doing, and every day we came here and we got stuff done while having fun. So it just became something we wanted to do every day.
I didn't have any experience with programming or EV3s before this, but I just came in and I jumped right in and I learned so much and I had so much fun. And it wasn't even uncomfortable because the people surrounding me was just a great environment to be in.
It might seem hard at first, but once you know what other stuff you can do, like the research, the building, the programming, then if you can find that spot you fit in best, then you can help your team out a lot more.
With me, I feel like it changed who I was, to say. I was definitely a follower, not a leader, but now I feel like I'm more confident to be a leader, and I think every student, no matter where you live, should be able to experience that with a group that they can learn to trust.
So the Galactic Farmers came all the way from Elmira Cooley Heartline. It's about a 2-hour drive over. And it's a small district, and they've come along with Shauna Shermer and Susan Douglas. Shauna is the superintendent of the school district, and Susan is their teacher. And I want to give the Galactic Farmers an opportunity to tell you why or how they feel about their awarding.
So this award meant a lot to all of us. Since we're all from a small community, we don't get a lot of opportunities like this in the STEM programs. So just having us being recognized for this award has really opened all of our eyes, and it's something we want to continue to do. And we want more STEM programs involved in our lives because it's meant so much to us, and we get a whole new world of opportunities that we didn't know we had before this opportunity came to us. We were very happy we got this award because I hoped it inspired other teachers and other students to start STEM programs in their school, because the STEM program is slowly going away. And doing this showed me that STEM projects are like really fun, and that other students should really get into STEM projects.
So, as it said in the video, we really did want to represent our community, and we wanted to prove that a small town can do huge projects and succeed into it, even if everyone assumes that small towns can't do it.
To me, it kind of was like something that I could do at first, but like how Katie said in the video, it kind of clicked in a few weeks. I think that's kind of how it should be for kids that haven't experienced this. They should be able to do something like with farming or technology to experience this type of
I think it was great that with this challenge, the Apollo challenge, it was more about like space and the moon, that we were also able to represent our community in agriculture while also using the technology part of it.
I am truly very proud that I got this award. I feel like I actually accomplished something, and I am actually very proud I'm representing my culture, because I honestly, our culture is about farming. Of course, you know that, but I'm really, really very proud. So I'm really proud of the students because of what they achieved. And the neatest thing was when we started the project, there was part of the criteria was to represent your community, but they could decide what that was. They could decide if it was just the community of themselves as students, as middle school students, and they chose to represent the agricultural community, which the effects of that were so powerful to not have anticipated. Often in schools, the dollars are very competitive. Extracurricular activities compete with sports dollars. It sometimes is hard to generate the funds that you need to provide some of the technology. And small schools are also very much about sports. That's really what drives the community, gives them opportunity to gather. And what this did was, from the onset, was really engage the community dialogue about what was happening in our middle school. And the incredible support that came from that, because these students were looking at agriculture as we had to develop a payload. Not only did they dress like farmers, but they developed a payload that had to do with a new grain that we're raising as food grade barley. And they chose to take that to the moon and leave it because of how barley, the nutrient values, and also the worldwide use of barley over wheat, which is traditionally what's in our area. And so their choices to do that, and then they built their projects around that, really excited and engaged our three communities, and even outside our region. The competition that they participated in was over in July, and I still get questions about how did the competition go, or what are we doing now. I think the component as an educator that's really powerful is that as a teacher, you can ask me what I think about STEM, and I would tell you, and it matters what teachers say. But when you could hear it through the student voice, when you can hear what their experiences are and how it changes their environment with three-dimensional STEM learning, they're not learning it from reading a report or watching a video about somebody else doing it, but really they're doing it. They're in a constant engagement with a device or with a science, and that's what it takes learning to a whole different level. And so it's their voices that really matter. You know, it really should drive what we do in education, is these kind of experiences. So thank you for the opportunity.
Who you are, because they're going to stick around for lunch as well, and we're hoping that you'll engage them and ask them questions. They would love to learn about you, and I'm sure you would love to learn a little bit more about them. Do you want to introduce yourselves real quick before? Yeah, go ahead. Yeah.
Okay. So I'm Nathan Hinkle. This is Max Carell. That is Beth Okamoto. That's Kaylee Elder. That's Katie Murray. That's Christine Keeley. That's our principal, Miss Shauna Schmier, and that's our teacher, Miss Susan Douglas.
Well, with that, I am going to, if it's all right with Jean, I'm going to go ahead and dismiss us for lunch. We've got 30 minutes I'll put up on the clock. Lunch is going to be in the patio over here. I really do hope that you'll engage some of the scientists that presented this morning, as well as the students that are here to tell you about their work. And we'll come back at 1 o'clock.